Good morning or afternoon, I should say.
I do the same thing, Tiffany. Good afternoon. Yes. Welcome back, everyone. Just give a moment for maybe another one or two folks to log in. Okay, great. I think I'll get started. So hello and welcome back. And I hope you're doing okay. And I am aware of some challenges in our communities because of the torrential rain that we had yesterday. So did anybody have a problem?
It hit pretty hard here. I'm from Oxford. So
Okay. But no problem with your home or anything like that?
no, thank God. But probably like 5 minutes from here, a lot of houses were ripped apart.
Oh dear. Okay. Well, I'm
Yeah.
glad you're okay. And so I'm glad everybody seems to be okay. I am aware of at least one student in my class that had a flooded basement, and unfortunately, the flood actually got a hold of the student's computer that they need to take the course. So she has no computer now. So we're struggling to find a solution for her. So things do happen, and I hope you and your families are okay. All right, well, I'm going to finish the Spanish flu story and get into chapter 1 legitimately with some notes. If there's any time left over, I will start chapter 2. Before I do you have any questions about the material that was presented last time in terms of your syllabus or course expectations? Okay, so you may have already noticed that I did send an email about an hour and a half, 2 hours ago with some files for lab. It is not necessary that you pre-read those files before coming to lab today, okay? In fact, I kind of intentionally waited to send them late, okay? But I just wanted to know that, check your email after class before we get into lab. You'll find an email message from me with some files that will be important for lab later this afternoon. Okay, so let's see here. We were talking about the worst pandemic in recorded human history, which was the 1918 Spanish flu. Would it be correct of me that I kind of stopped the conversation after we had mentioned the cytokine storm? Is that true?
Yes.
Okay. All right. So lab today, just checking in the yes, thank you, Samantha. Lab today is at 2 o'clock. All right. So yes, just continuing with this Spanish flu story. Let me ask you this. I'll type it into the chat window. Where do you think the Spanish flu originated? Okay. I mean, it certainly seems reasonable to say that the Spanish flu originated in Spain, right? It's kind of like asking, well, who's buried in Grant's tomb? Well, it would seem reasonable that Grant is buried in Grant's tomb. But you know what? History is odd, humanity is odd, and life is stranger than fiction. As far as forensic historians can tell, for people that have done the detective work on this, it would appear that the 1918 Spanish flu did not actually originate in Spain. It originated in Haskell County, Kansas, United States of America. Okay. So you say, well, what the heck is that all about? Why in the heck is it called the Spanish flu rather than the Kansas flu? Why is it not called the Kansas flu or the American flu or whatever it was? And I draw your attention to those sorts of things where you need to think beyond your narrow-minded focus when you're talking about a topic in school. Just because we're talking about the flu right now, don't ignore the fact that there were many other important events happening around the world in 1918. And in fact, I think one of you even mentioned it last last time. So let me ask you, feel free to unmute your microphone. What massive global event in addition to the flu was taking place in 1918?
The War of 1918?
Never mind.
Okay, I understand what you're saying. Yeah, no, not the War of 1918, but you did hit on war, right? It was World War I. World War I was taking place in 1918, okay? And so think about it. Even today, our societies and governments really aren't any better than they were in some regards to 1918, right? Isn't it generally true that whatever's happening in the world, governments like to control the flow of information, make sure people have certain information, not other information, sometimes even propaganda. So what I'm telling you is that during the 1918 flu pandemic, the United States government would not allow newspapers to publish information about the flu. And the reason for that, and other nations around the world were also not allowed to publish in newspapers, the primary media of the day, were not generally allowed to report on disease outbreaks of Spanish flu. And the reason was that so many people were dying during the war effort, that we didn't want to depress everybody and tell them, oh, by the way, there's also a bunch of people dying from this unknown disease over here. Okay. So the reason it became the 1918 Spanish flu is because the Spanish government was the first government to sit back and say, you know, this is kind of silly. This is a little ridiculous. People need to know that there's something going on here, because people aren't stupid. We need to admit this. We need to recognize it. We need to get the word out to help people in whatever way we can. So while the forensic history of this disease suggested that the flu probably originated somewhere in the United States of America, and again, Kansas seems to be the primary runner for the data, it became famous as a Spanish flu because the Spanish government was the first to publish and admit that there was something going on about this. Spanish government allowed Spanish newspapers to print information about what was happening. Okay, some other talking points here. The original causative agent, right? A causative agent is also known by a fancy name as etiologic agent, right? The etiologic agent, the causative agent of Spanish flu, was originally thought to be a bacterium that got its name because of an incorrect association with this disease. This bacterium today is known as Haemophilus influenza. Haemophilus influenza. This bacterium does not cause flu. It does not cause flu, but in 1918, the best medical advice of the day, from what limited research could be done on the cause of the disease outbreak, had pointed a finger at this bacterium. And the reason for that was when folks unfortunately died and succumbed to the disease, you know, in medical schools, they would take the person's body and they would do an autopsy on the person's body, and they would try to culture out various microorganisms from various body surfaces, including the lungs and respiratory tract. And frequently, but not always, one of the organisms that they could culture out of the lungs of persons that had died from the Spanish flu was the bacterium Haemophilus influenza. Okay, so that's how this bacterium was incorrectly identified as the cause of the flu in the 1918, 1919, 1920 area of time. Okay. Most likely, the reason that bacterium was prevalent in the lungs of folks that had passed away from the flu is because while the bacterium does not create flu, the bacterium does cause pneumonia. And you know, I mean, if you've been around people that have had flu, a very serious secondary concern that can happen is that you also develop pneumonia simultaneously. So your body is just really worn down. Let me ask you in the chat window, Have you ever experienced a really bad case of the flu? Okay. I mean, a case where you remember that it was quite unpleasant and you really probably don't want to experience that again. Okay. So notice that some of you are saying yes, and some of you are saying no. So God bless you, those that have been fortunate enough that the answer is no. And I can raise my hand and say, yes, I have. Many years ago, I had a really bad case of the flu, and it knocked me down really hard. So I don't wish it upon you or anyone, okay? All right, so today, of course, we're modern and sophisticated. We know things like viruses. You see, in 1918, nobody really knew what a virus was. Nobody really knew about viruses. There was some rumbling in the research community that there was something so small it could get through their filtration, their rudimentary filtration papers that they had at the day. So people were thinking, man, maybe there is something smaller than a bacterial cell, but we don't know what this is yet. Long story short, it wasn't until 1930 that the scientific community finally stumbled on the true cause of the flu, and they did so by studying an outbreak of swine flu in a herd of pigs up in Canada in 1930. So in 1918, humanity experienced the Spanish flu, worst disease outbreak in recorded history. It took another 12 years of investigation and research to lock it down that, oh, the flu is actually caused by a virus. And the original studies were done in cases of swine flu outbreak in Canada. Okay, so you know, I have to read my notes because I don't have a very good memory these days. World War I was going on. World War I was going on around the globe. World War I officially started August 1st, 1914, and it officially ended November 11th, 1918. The United States of America did not enter the First World War officially until April 6, 1917. The war itself made it very difficult to deal with this disease outbreak, okay? Obviously, at the time, if you're going to go to war, you need to train and equip your troops, which means you need to move men and women, but in 1918, it was more of the guys, need to move men and equipment to training camps where they can be equipped and trained in all this. So that meant they were shipping men typically by rail or ship in enclosed confined spaces for training camps and things like this. And the medical community of the day kept telling the United States Army and so forth, please don't do that. Please don't do that, because all you're doing is exposing the men to this unknown disease that we can't control right now. Well, you know how things go. The US military largely ignored that advice, and they continued to do that. And so many men and women did die from simply being conscripted into military service of the time. In fact, in the United States, at least, most of the medical personnel were conscripted into medical service so that, you know, they were training on military bases and taking care of folks. It meant that the local towns and communities in the states that had existed in America had very little medical personnel to care for people that were sick. So if you were sick, you were kind of on your own. There wasn't anybody that was, quite frequently, there was just nobody around with any medical training that was going to help you because they were all dealing with the war. Okay. Philadelphia, I'm sure you've all been to Philadelphia. Philadelphia was especially hit during the Spanish flu, in part because it was the location of a naval shipyard. During the week of October 16th, 1918, in Philadelphia, 4,597 people died in 1 week. 4,597 people died in 1 week in Philadelphia. Would it be fair to say that in whatever town you currently live in, if 4,500 people drop dead this week, you would be alarmed? Okay, so it was scary stuff, very serious stuff. Okay. The statistical records of history reveal to us that it's estimated that up to 40 million people died through combat in World War I. So 40 million people died by combat in World War I. And as you know from last conversation, we estimate at least 50 to 100 million people died from the Spanish flu. So more people died from the Spanish flu than died during the entire time frame of World War I. In Europe, Europe was not spared problems. Europe had great problems. Between June 1st and August 1st of 1918, 200,000 British soldiers that were stationed in France became ill with the Spanish flu. They became so sick that they could not be put into combat. And the point here is this, that the people that those 200,000 troops that survived, many historians say that they survived because they couldn't go into combat. In other words, getting sick with the flu actually prevented them from seeing frontline activity. Okay, so let's see here. I'll wrap up this story with a little anecdotal, quirky kind of piece of information here. Okay, for those scientists and historians that study this period of our world's history, there are medical records and documentation from doctors that treated patients in the day that not only listed traditional flu symptoms, you know, that we've all experienced, you know, runny nose, headache, fever, all those kinds of horrible things, but they also mention altered mental status. There was a lot of mentioning of altered mental status. And of course, if you have altered mental status, it suggests that the virus is in some way impacting the central nervous system. Okay, so at the conclusion of World War I, the American president, which at that time was Woodrow Wilson, went to France to be part of peace treaty negotiations between the French premier, his name was George Clemenceau, and the nation of Germany, which obviously was our opponent in World War I. Well, the French people had suffered tremendously during the war and saw a lot of combat on French soil. Obviously, there was no physical combat on American soil. So President Wilson had a different perspective on how we should treat the Germans than did the French premier. The French premier really wanted a very harsh peace treaty deal with the German nation because of all the things that had transpired on French soil. So there was disagreement between these two world leaders of how to negotiate with the Germans. During the peace treaty negotiations, Woodrow Wilson got sick. Guess what? He got sick with Spanish flu. He went back to America to convalesce, to rest, to get better. And there are people that worked in the White House during that period of time, and through their memoirs and notes that they have written, they suggested that Woodrow Wilson kind of was acting really wonky, really paranoid during that period of time. And of course, you can't help wonder if the Spanish flu virus altered his mental status at some point. Well, long story short, Woodrow Wilson went back to France after he recovered. And when he went back to France, he conceded his position to the French premier and said, you know what? Okay, let's do it your way. We will get a harsh peace treaty with the Germans. We'll do it your way. So President Wilson gave in to the French position. Well, if you're a student of history, you know that it was some 20 years later, and through the rise of the Third Reich and Adolf Hitler, that World War II began. At least some fairly significant body of historians suggest that the conditions that created World War II was the harsh peace treaty deal of World War I. So you have to ask yourself, could a lowly virus like the influenza virus have played a role in creating the first World War I treaty by causing President Wilson to concede his position to the French premier, thus setting up a harsh peace treaty after World War I, creating 20 years of unrest and discomfort in the German people, allowing the Third Reich to rise, and away we go with history of World War II. We'll never know, but it's kind of interesting to think about that. Okay, so I usually talk about, before coronavirus hit us, I usually talk about, well, what are we going to do now if we have another pandemic like Spanish flu? Well, so far, coronavirus is not as bad as Spanish flu, although it's certainly bad enough, right? There's still a lot of suffering going on. And, I used to make a point of, that before coronavirus hit, if you were to count up all the ventilators across all the hospitals in the United States of America, you might have counted about 200,000 ventilators. And so the question becomes, if you have 300,000 people sick and needing a ventilator, what's going to happen, right? Now, we have increased our inventory across the nation of ventilators. I honestly don't know how many we have now, but I know through certain emergency protocols, manufacturing lines at Ford and other big companies were put in place to build additional ventilators. So I do think we do have more ventilators today than we did before COVID, but I don't know how many we had. But at any rate, I put this to you as students that are probably going into the medical arena, right? You're going to be putting yourselves in harm's way if you're not already putting yourselves in harm's way. Think about it. As a society, we kind of deal with a low level of annoying diseases every day, every week, every month, every year, such as cancer, right? We expect a certain level of cancer in our society and we just get used to it. Oh yeah, Henry died at 72 of colon cancer. He was 72, cancer, okay. But you have now seen, you're living it now, and you're hearing it from me from the history book, Spanish flu, just how absolutely devastating an infectious disease agent can be, right? It ripples through society and it continues to mutate into different variants, and it keeps going, and we don't yet see an end to this. Okay. So, do you have any thoughts or questions about the 1918 Spanish flu? Okay, so thank God our current situation is not as bad, but it's certainly bad enough, right? Certainly bad enough. Okay, gang. So that's a little history lesson of infectious disease. That's not too far away, right? 100 years ago, none of us were alive, but there's plenty of historical documents to tell us that things were really nasty. I mean, there's letters written from families where mom and dad would go to bury a child that had died from Spanish flu. They'd go to the funeral, and when they got home, their second child had died from the Spanish flu. Okay, this was really bad stuff. We don't wish this upon our communities because it's extremely harsh. Okay, so this is what we're dealing with in microbiology. All right.
Dr. Allen,
Yes.
I have a question quick. Is there any possibility of the Spanish flu coming back?
It's really difficult to predict the future. I will say that some studies have been done with modern technology, with modern DNA technology. Some studies have been done in the research lab on the Spanish flu, and scientists are working hard to figure out what made it so absolutely devastating. I don't really know what the conclusion of those studies have been, but if I were to guess, my suspicion is what made the Spanish flu so devastating is that it was very good at infecting cells. And with coronavirus, we started out with the Alpha variant, and now we currently have the Delta variant we're dealing with. The Delta variant has mutated in several months from the Alpha variant and has changed some of the amino acids on its surface proteins that allows it to get into our cells better. That's a big problem. So I think Spanish flu was probably so bad because it could do something similar. Of course, having a population that would ignore medical advice to not congregate into groups and things like that made things a lot worse. And of course, there was no vaccine technology in 1918. They didn't even know that viruses existed. So how could you create a vaccine against it? Just was not possible. So there were some things about that moment in history that made the flu super, super bad. If it happened today, if it resurrected today, I don't think it would have quite the severity as it did in 1918, because hopefully we're a little bit more reasonable in our thinking, and we do have vaccine technology today. So hopefully we can mitigate it better. But could it come back, or some very slightly close cousin to it? I don't see why not. Okay.
Okay,
Other
thank you.
questions? Okay. All right, gang. So, we don't need to be paranoid about infectious disease, but we need to be very serious about it. We need not to be silly. Okay, I'm going to share my screen and bring up notes. As I said, I'm not going to list the entire chapter out for you. It's a history chapter. Hopefully you've read it or are in the process of reading it, and hopefully you find it rather interesting. There's certain pieces of information that I want to get into your notes from chapter 1, and we'll start here. We'll start here with this question. What is microbiology? Microbiology is the study of microorganisms, or we might say microbes, okay? Well, that doesn't mean too much if we don't know what a microorganism is. So let's talk about that. Okay, what is a microorganism? Okay, a microorganism, okay, I'm going to give you a definition that I'm going to give you some pause. Microbiology historically has been that subdiscipline within biology that if you don't know where to put a topic to study, you kind of just dump it into microbiology, which means it's kind of hard to give you a universally correct definition of microorganism when so many different topics get thrown into the field of microbiology for study. But I'll give us a good working definition to get started with here. Okay. All right, let me get you guys up here. Okay, so a microorganism is any cell or virus. And that's probably one of the biggest things you can appreciate. If you have not previously appreciated that a virus is not a cell, that's super important to appreciate, right? A virus is not a cell. Any cell or virus that is too small to be seen with the naked eye. Okay. Any cell or virus that's too small to be seen with the naked eye, any cell or virus is too small to be seen without the aid of magnification, that would qualify as a microorganism. Okay. Now, there are some problems with that definition, right? Because a human red blood cell is certainly too small to see with the naked eye, and yet we don't traditionally talk about red blood cells in the field of microbiology. We deal with that in human medicine. So there's a little bit of fluff and fuzz around how we define things, okay? And so I'm asking you to be flexible on your appreciation that there's not a universally hard and fast definition that satisfies every circumstance that you could bring up. And I will say that probably, I don't know, 15, 20 years ago, a bacterial cell, a bacterium, was identified off the coast of Africa that in fact was so large you could see it with the naked eye. All right, so even bacteria on our planet, some species of bacteria can get large enough that if you have really good eyes, okay, you can see these things without additional magnification. Okay, so let's talk about how we name microorganisms. How are microbes named? Okay, and so I don't want to get into a big conversation about systematics. Systematics is the way of naming and organizing different life on the planet. This course is not about that, and I find that to be very dry material. But you name microbes just like you name an animal with a genus and specific epithet. Okay, a genus and specific epithet. So let me give you some examples. Okay, let's just take our own species, right? Homo sapien, okay, that would be humans, right? So let's go right to microorganisms. Let's talk about E. coli bacteria, right? E. coli bacteria is found naturally in our large intestine, our colon. And so we would write it out this way, Escherichia coli, Escherichia coli. Okay, let me give you another one. That would be the bacterium that was misidentified as the cause of the Spanish flu, Haemophilus influenza. Okay, Haemophilus influenza. And we can go on and on and on. Okay, so obviously this is humans. This is bacteria. This is bacteria. And I'll give you one fungus here. Okay, there's different categories of fungi. One category of fungi is yeast. Okay, so let's talk about baker's yeast. That is Saccharomyces cerevisiae. Okay, so if anything in this course, you're going to run into, let's see here, I need to have an E on the end of influenza. You're going to run into some big, long names that are kind of intimidating. Okay. Let me say this. Generally speaking, you know, if I'm talking about a topic in the class and I want to refer to Escherichia coli, I'll probably say E. coli. It's just easier, right? Generally speaking, I won't be a stickler about writing out full names. I won't be a stickler about getting perfect spelling on things. I won't generally be a stickler about those things unless I give you an assignment or an exam question where I specifically say, you need to write a full, complete, proper scientific name for the microorganisms that are part of this conversation. There will be one essay on your first exam where I do require you to write full, complete, properly formed scientific names, okay? Because you need to know how to do that. Now, the thing about the genus and specific epithet is, you notice a couple things, right? The genus is always uppercase, right? It's always capitalized, all right? And the specific epithet is always lowercase. Okay. Additionally, the scientific name that you write should either be in italics or it should be underlined. So the scientific name should be in italics or underlined, not both. If you were to italicize and underline, that would be technically incorrect. That's not how the field does it. Okay, so I'm going to go up here and put in, I'll make these things italicized for us in our notes, okay? Now, I won't always take time to do that because I'm just trying to talk about a microbe and get on with the conversation. But right now, the point of our conversation is that you can recognize a scientific name, a genus and specific epithet, and that you could potentially write it properly if you had to do so. You know, many of you are at the early stages of your career, but there's going to be a time in the future where you're going to be the teacher, you're going to be the supervisor, you're going to be the department head, and you might have to write some technical documentation, instructions, policies. And of course, to act and look like a professional, it's going to be expected that you can write a proper scientific name for a pathogen. Okay. All right. Okay. Do you have any questions so far?
Actually, I had one question. I was just wondering if these notes would be up on D2L.
I probably won't put them on D12, Samantha, but I will send them to you through email.
Okay, that's cool. I was writing them down just in case, but okay, thank you.
Yep. No, it's let me, if I didn't say it last time, I'll say it again. I very much encourage you to take your own class notes during class time, because if you take notes, you're in the game, you're thinking about the topic. That helps you understand and helps you succeed. Don't just rely on my notes, because there might be times I say things that I don't write down, but I will send these to you. Okay. All right. So
You're gonna send them to everybody. I'm sorry, because I'm also writing them down, but I like to make sure that I don't miss anything.
yes, Tiffany, I pretty much after every lecture class, I will send the entire class an email with a link for the lecture recording and also the notes. So you will get them. Anything else? Okay. So so we've got started here. We know what a microorganism is. We know how to name a microorganism and write it out properly. What I want to cover next is cataloging the various types of microorganisms that we typically deal with in a class in a field of microbiology. And if you do have your textbook available, I'm looking at pages 4, 5, and 6. Pages 4, 5, and 6. So types of microorganisms typically studied in microbiology. Now, this list here is a pretty good list that your textbook has put together. It's not technically exhaustive and completely comprehensive, but it's a very good starting point. Okay, so first off, let's start with some definitions here. Okay, first off, when we talk about various kinds of cells, we know that there's basically two broad categories of cell life on the planet, right? We have prokaryotes and we have eukaryotes. Okay, these are labels that we ascribe to different kinds of cells, right? There's prokaryotic cells, eukaryotic cells. You would not say prokaryote in the context of a virus, because it makes no sense, because a virus is not a cell. There are not, you wouldn't call bacteriophage lambda prokaryotic. That would just make no sense. Okay, so prokaryotes and eukaryotes are referring to types of cells. So this is a type of cell without a nucleus, right? A prokaryote is a type of cell without a nucleus. There might be some other differences that we could highlight, but if you think about life on our planet, and I don't want to get into any philosophical disagreements, it really doesn't matter whether you believe in religion or evolution or kind of both. It doesn't matter. What I'm pointing out is that as far as we can scientifically say, life on our planet started evolving about 3.5 billion years ago somehow. And so through 3.5 billion years of evolutionary history, there's been all kinds of cells. So there's been cells that are prokaryotic and eukaryotic, and there's probably a cluster of cells that are somewhere in between our artificial designations of prokaryote and eukaryote. If you see what I'm saying, there's a transition zone of cellular life that could be prokaryotic, could be eukaryotic, because it kind of spans the distance between those two terms. Okay. So prokaryotic cell is a type of cell without a nucleus. Eukaryotic cell, a type of cell with a nucleus. Okay. All right. So I'm going to talk to you about prokaryotic cells first, which we typically talk about in microbiology. And bacteria are right at the top of the list. Okay, bacteria are prokaryotic cells. They're underneath the category here of prokaryotic cells. They do not have a nucleus. And so the examples I could give you, I already did give you. I gave you E. coli, I gave you Haemophilus influenza, right? If you look at page 5 of your textbook, picture A, you have a picture there of Haemophilus influenzae. Again, it causes pneumonia. It does not cause influenza, okay? And by the way, now most of the photos and pictures you see in page 5 are artificially colored. You know, the Haemophilus influenza do not have a aquamarine blue color to them. That's computerized colorization to make your textbook look fancier than it should be. Okay, cells don't really look like that, okay? So, Haemophilus influenzae. Let me give you another one, Vibrio cholera. Vibrio cholera is the organism that causes severe dysentery and other diarrheal diseases, particularly in third world nations. Even today, it causes a lot of problems. Okay. So Haemophilus influenza, Vibrio cholera would be examples of bacteria. You know, certainly from a medical perspective, you're going to see cases. I mean, if you're working in a family physician's office, you're going to see patients every day that are having some kind of problem with a bacterial infection, or, you know, in the hospital, whatever, nursing homes, right? You're going to see it every day. Okay, number 2, second category of prokaryotic cells is something known as an archaeal cell. Archaeal cell, archaea. And these kinds of cells are prokaryotic. They are genetically a little bit evolutionarily distant from bacteria, but they're closer to bacteria than they are eukaryotic cells. Like archaea are more similar to bacteria than they are to human cells. Archaea are more similar to bacteria than they would be to a yeast cell like Saccharomyces cerevisiae, okay? So when we have these different categories, organisms are placed into a category based on slightly genetic differences, okay, in their genetic sequences. Okay, so archaea, let's see if I can find, hold on a sec, I have a page number written down in my notes here. Let's see if it's still valid. No, of course it's not. Okay, all right. Well, let's do this. Let me just do a Google image real quickly on this. Okay, so Google Images is fantastic for our course, specifically when we're forced to be online. So I would encourage you to use Google Images to look up what microbes look like at various times. Here's one, Sulfolobus sulfatacaricus. Okay. Here's what the beastie looks like. All right. Okay. Doesn't look terribly remarkable. Again, it's, you know, artificially colored. But at any rate, this organism was originally discovered around a volcano in Italy, And if there's anything that kind of distinguishes archaea from bacteria, one of the things that we take a look at as humans is we often find archaea in environments that would be very inhospitable to human life. Okay, so archaea often get the designation extremophiles because they live around volcanoes, around deep sea ocean vents where there's a lot of pressure and acidity, temperature, that kind of thing. So they're kind of extremophiles in the sense that they live in areas that are kind of hostile typically to animal life. Okay. So let me give you this example here. Sulfolobus. Sulfaticaricus. Sulfolobus sulfaticaricus, found originally in a volcano in Italy. Okay, so I'll put down extreme mobile. Okay. All right, gang. So actually, bacteria and archaea are the only two categories or subdivisions of prokaryotic cells, typically that we study in the field of microbiology. Okay, so bacteria and archaea are it. Are you going to encounter archaea on a daily basis in a clinical setting that are making people sick? I would say no. Okay, I wouldn't be willing to say you won't ever encounter a case, but I don't think you're going to routinely encounter it, and you might encounter it in ways where you're not even identifying it. I mean, let's be honest. How many times have you been to the doctor because you were sick with strep throat or something that seemed like it was some form of bacterial infection? And, you know, the medical office never bothers to try to culture or identify what the pathogen is. They simply throw a pill at you and say, take it for 5 days, and hopefully it goes away, right? You're going to find that I'm not too happy with the medical community because of practices like that, but it is what it is. I can't stop it. We don't want to bother to measure what the hell's going on. We simply want to make an educated guess and hope things work out okay. I have a good friend that's now on his 3rd week of different antibiotics because what they've given him so far is not working. Of course, they never tried to identify it. So at any rate, here we are. Okay, so those are prokaryotic cells. Any questions so far, right? We're just whetting your appetite for things. We're going to be going into the physiology of these things much more throughout the entire semester. Samantha, you had mono and they sent me to 3 specialists before they tested it. Yeah, I'm sorry about that, right? We have a culture, you know, well, I don't want to get myself in trouble. We have some problems in the medical community, okay? So anyway, and part of the problem is who's going to pay for it. Part of the problem is who's going to pay for it, okay? So at any rate, eukaryotic cells, okay? So bacteria and archaea are 2 different types of microorganisms that we study in microbiology. Eukaryotic cells. Here's some eukaryotic cells. We have, let me continue my numbering, okay? So I have 1 and 2. These are prokaryotic cells. I'm going to continue with 3 as long as you appreciate that 3 is the first eukaryotic cell that I'm listing, okay? So 3 is fungi. And there are various types of fungi. There are various types. We have yeast. Okay, we have yeast, and I already gave you an example of that. That would be Saccharomyces cerevisiae. I'll type that out again. We also have mold. Okay, now your textbook on page 5, panel B there shows you mucor. Shows you mucor, which is, as they say, a common bread mold. Okay. Question to you coming in the chat window. If I could type properly, this would go well. There we go. Have you ever eaten moldy bread or any moldy food? You really haven't lived until you have. So I encourage you to do it this Labor Day weekend, okay? So, okay, very good. So, I mean, my point is that you know what mold kind of looks like on bread, or you know, you might have some leftovers in the refrigerator that you just let go too long and you open the lid to your leftovers, like, oh yeah, that's nasty. We're going to get rid of that. Okay. So when I was in doing my graduate work in Maryland, I went down to the campus store because I was so hungry. I'd been there a long time and I got a pack of Twinkies. No, Crimpets, butterscotch Crimpets. I proceeded to pound those down, and in the last moment, I realized they were all moldy. I was like, oh, this is disgusting. Okay, so mold is a fungus. And I'm going to give you the example of arguably the most famous mold in the history of humanity. The mold that is the original source of the antibiotic penicillin. Penicillium chrysogenum. The source of penicillin. Okay. All right. So notice I'm not taking time to italicize these because that just takes more time away from my notes with you. But if you were writing them properly, everything would be italicized or underlined. Okay, so we have yeast and molds, which are different subtypes of fungi. We also have mushrooms. Okay, so please don't tell me if you've ever done psychedelic mushrooms. Okay, don't tell me that. I don't want to know that. Okay, but let me ask you this. Let me ask you this in the chat window. Okay. When you order pizza from Papa John's, do you order pizza with Agaricus bisporus on it? Well, you should. Okay. Agaricus bisporus is the most common mushroom that you will find at the grocery store, and you will find on things like pizzas. If you ask for mushrooms on your Domino's pizza, most likely it is of the variety Agaricus bisporus. Okay. And there you have it. Okay. Okay, folks. So those are the three kinds of fungi: yeast, molds, and mushrooms. All three of them are a subtype of fungus. Okay, so there's a fungus among us. Okay. All right. We will talk about fungi, athlete's foot, all that kind of good stuff later on. Okay. Next eukaryotic cell category is protozoa. Okay, protozoa. Okay, there are many examples of this that I could give you. I'll give you, well, okay, let's take a look. Page 5, picture C there gives you one example, right? The old classic amoeba that you might have studied in biology class in high school. Okay, so the full term for that would be amoeba proteus. Okay, that's a type of protozoa. Another example that you're commonly aware of that you probably studied in school is paramecium, right? Now the full name, there's many different specific epithets for paramecium. There are millions of types of protozoa on our planet, millions of types. Nobody knows them all. Okay, but here's one common one that's studied in school: Paramecium aurelia. Paramecium aurelia. Okay, so maybe you've had some experience in biology class years ago. Okay, can you get parasitic pathogenic protozoa that can be a problem in the clinic? Yes, you definitely can. You definitely can. Okay, I don't know how much, you know, we all have our lives and we're busy, but every year, invariably every year in the spring and summertime, I hear about a case of, it's often unfortunately a child, but it could be anybody, that gets a case of a parasitic amoeba infection from an amoeba called Nagari fowleri. And what this thing is, it is basically a brain-eating parasite, okay? So what happens here with some of these protozoa that are toxic to animals and humans is, in the case of children, they might be out playing in a muddy puddle or a lake, stream, waterway of some kind, and they're splashing around, right? They're splashing water into the face. And in that pond water or that puddle water, there is this parasitic protozoan. It gets into the nostril. It goes up and it's able to penetrate the blood-brain barrier around the brain. And once it gets in there, it starts consuming the neuronal tissue. It is 99.9% fatal. We can't deal with that. By the time you know that this is happening to your child or to your mom or dad or whoever has it, by the time the medical doctors know what's actually happening, it's almost always too late to do anything about it. I'm only aware of one person that survived it, and I think they survived it by putting the person into a medically induced coma. Okay, so there are some nasty things out here. And again, I don't want to scare you in this course, but I need you to be real, right? We don't want to be foolish about how we do things, okay? There is nasty stuff out there. So protozoa is another type of eukaryotic cells, one of the major types of cells, types of microorganisms we study in microbiology. Okay, next category, number 5. Okay, 5 would be algae. Okay, algae, a picture of algae is shown to you on page 5, picture D. It's the alga volvox. Okay, algae, these are plant-like. They are plant-like in nature, and by plant-like, I'm trying to tell you that they have chloroplasts. Okay, they have chloroplasts. And an example, I'll use the example from your book. This is Volvox aureus. Okay. Are you going to find toxic algae? Humanities, science is aware of some toxic algae, but it's pretty uncommon to come in contact with it in the clinic. I mean, you would have to have a person that's been doing some rather unusual activity to be exposed to it. So generally speaking, you're not going to deal with toxic algae in the clinic. That's not to say it will never happen in your career, but it's not going to happen on a daily basis. Okay. Okay. All right, gang. So let me say this. Categories 1, 2, 3, 4, 5. And I'm going to put another one in here. Okay. Hold on here. Number 6. Number 6 are multicellular animal parasites. Okay. You'll notice on page 6 of your textbook that this category is listed, multicellular animal parasites. And you'll notice the first sentence of that paragraph on page 6 says this: although multicellular animal parasites are not strictly microorganisms, they are of medical importance and therefore will be discussed in this text. So there you go, folks, right? We talked about what a microorganism is, and I said you need to be a little flexible on your definition because a lot of times we throw things into the field of microbiology that we don't know where else to put them. So multicellular animal parasites are not strictly a microorganism. I mean, because it includes things like flatworms, tapeworms, generally things called helminths, okay? And, a flatworm, a helminth inside your digestive tract could be 3 feet long. It could be 3 feet long. In fact, I think the Guinness Book of World Records for the longest worm that was pulled out of a digestive tract is about 6 feet. So obviously you could see a 6-foot long worm. That's not a microorganism by any traditional sense of the description. But we do study things like worms and multicellular animal parasites in the field of microbiology. Okay, these are not strictly microbes, but are studied in microbiology due to their medical importance. Okay, let me see if I can find a page number in your book to look at here. Okay, oh, beautiful. Okay, if you want to see some gross things, turn in your textbook to page 752, 753, 752, 753, and you're going to see some worms here, right? Figure 2523 shows you a cyst inside a person's lungs. That cyst is made from the worm that we call Echinococcus granulosus. Echinococcus granulosus, you see an ancylostoma hookworm on figure 2524, and you see asparagus worms on figure 2525 again, right? So some really gross things. So if you've ever, I know some of you probably have small animals, dogs at the house. Have you ever had to deal with heartworm treatment for your dog? Okay, so think about the heartworms, right? That would be of medical or veterinary importance, which is something we study in microbiology. Okay, so let me put down as the example here, Echinococcus granulosus. Echinococcus granulosus would be an example of a multicellular animal parasite. Okay, so now, double asterisk here. Items 1 through 6 in the list above. Items 1 through 6 in the list above are all cells. They're all cells, or they're made of cells, right? An alga is a cell. Now, protozoa is a cell, fungi is a cell, archaeobacteria are cells. Okay, a multicellular animal parasite is a multicellular structure, a multicellular organism. Items 1 through 6 in the above list are cells. And so you can use the normal scientific naming convention that we started today with, genus and specific epithet, to represent these cells. Okay. Now we're going to go into something else that's also studied in microbiology, but it's not a cell. And therefore you cannot use the genus and specific epithet naming process or structure to name things like viruses. Okay, so we're going to go now to the next thing, which are viruses. Okay, viruses are not cells. They are not cells. Okay, so in fact, it's so important that you appreciate they are not cells. I'm going to type it again. These are not cells. Okay, these are not cells. And you cannot use the normal naming system for viruses. Viruses are not named with genus and specific epithets. Okay, we will talk about how viruses are named in chapter 13. The reason viruses are not named with genus and specific epithets is because they mutate too rapidly. They're constantly changing. They're constantly changing. And because they're constantly changing, as soon as you name it, the thing that you name changed, and all of a sudden it's not what you think it is anymore. So this is the reason we don't use genus and specific epithets for viruses. Typically, you will refer to a virus by a standard name like HIV-1, HIV-2, HIV-3. And that designation of HIV-1, HIV-2, HIV-3, or coronavirus alpha, coronavirus delta, those designations will refer to a very specific and precise sequence of DNA or RNA that's in a computerized database. So that HIV-1 has a very precise nucleic acid sequence. HIV-2 has a different but precise nucleic acid sequence. I'm talking about DNA or RNA. Okay. Viruses are not named with genus and specific epithets because they mutate too rapidly. Okay, they mutate too rapidly. All right, so we are going to do an entire chapter on viruses in the future. It's chapter 13, and it's one of my favorite chapters because as irritating as viruses are, I mean, they make us feel horrible at times, and it's ravaging society right now. They are just so cool in what they can do. Okay, so at any rate, an example for virus, take your specific virus that you're friends with, right? We talked about influenza virus. Your book talks about Zika virus, right? Zika virus, HIV, coronavirus. So pick your favorite virus and you can put it in there. Okay, once again, the picture of Zika virus on page 5 of your textbook is artificially colored so you can see it. Zika virus is not a glowing orangish red. That's just completely artificial. Okay, so I'm going to list number 7 as viruses. So let's put, for the category here, I'll put this, acellular. Acellular, and by acellular I mean things that are not cells, things that are not cells. Okay, please forgive me for saying this, but at least one of you is going to tell me on your future exam that a virus is a cell. At least one of you are going to tell me that because it happens every semester. Okay, viruses are not cells. It's a total different ball game with viruses. Okay. All right, so these are basically the 7 categories that your textbook presents: bacteria, archaea, fungi, protozoa, algae, viruses, multicellular animal parasites, right? I just read right from the book, so I've organized it a little bit differently. Folks, I'm going to list one more for you. One more thing that your book doesn't intentionally put here, but I think is worth your conscious energy here, and that would be prions. Okay, so let me ask in the chat window. Okay, what is a prion? What is a prion? Anybody run across this before? I know you can Google it. You don't need to Google it, but if you happen to know, do you know what it is? Okay. All right. Well, folks, prions are misfolded proteins that create disease. Misfolded proteins that create disease. Okay. All right. So, you might have intellectual curiosity about microorganisms in general, but typically we study things because at some point in history, somebody's been harmed by them, right? So we know about bacteria causing disease and fungi causing disease. We certainly know about viruses, but prions can cause disease as well. And prions are not cells and they are not viruses. They are simply proteins that have adopted the wrong shape. They've adopted the wrong shape. And when they adopt the wrong shape, they create disease. So let me give you some examples of diseases caused by prions. And this will just be a quick list here. Okay. How many of you have heard of mad cow disease? Mad cow disease is a prion disease. Okay. Now, most of us might chuckle a little bit and say, well, too bad for the cows. But guess what, right? If cows have this infection, is a farmer going to be able to sell his or her beef? Not going to be able to sell it. All that cattle's got to be destroyed. Going to have devastating consequences for the agricultural economy and devastating consequences because we can't feed our people. We can't feed our children because who's going to buy meat if there's even a suspicion that there's a prion-based situation with the meat? So mad cow disease is one. You may have heard of sheep scrapie. That is another prion-based disease. I don't know if I have any hunters in the virtual room, but maybe you've heard of a disease that hunters care about called chronic wasting disease in deer and elk. And then there's one more disease, which is a neurological condition that occurs in human beings called Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. Most prion diseases, certainly that I'm aware of, are diseases that heavily impact the nervous system of the animal. Okay. So, are you going to come across a prion situation? Well, it depends where you live and work. You might, if you live particularly in a rural community where agriculture is a big deal, you might deal with an outbreak of some prion-based disease in the farm community, which then has to be taken very seriously. Okay. All right, folks. So I think that's about all I'm going to lecture on from chapter 1. Okay, so do you have any questions from our conversation today or from anything you've been reading? Okay. All right. Trying to
Dr.
debate.
Allen, I have a question quick.
Okay.
In the syllabus, it says, for study habits and things like that, that vocabulary is very important. Now, do you like the book's definition of like some of the vocab words? Or, I mean, I know that you said that, it would be best to like write one or 3 different ones, right? The one you give, the book gives, and then ours. But is it different? Like, I'm not sure. I haven't really.
So it's a tricky situation, Rachel, because what I want for you is that you come across or develop a definition that you understand that is technically correct. A definition that is technically correct that you understand is what I desire for you. And so it's hard to say how you will accomplish that. You might accomplish that from the textbook definition. You might accomplish it from the glossary in the back of your book, right? You do have a glossary back there. Maybe that's what you want. Alternatively, many words in life have alternative or slightly subtle differences in meaning based on context, and that's where lecture and conversation together can tease out a nuance that maybe a textbook definition doesn't have. So the point is, try to give yourself a definition that seems technically correct with what you're reading and hearing in the course, and which you sincerely understand, not that you memorize and regurgitate. I'm not interested in that. I'm interested in, do you really get what the words are telling you?
Okay, thank you.
Okay. All right, so there's 1 minute left. I don't think it makes too much sense to start chapter 2. So what we will do is certainly hit chapter 2 very heavily after Labor Day, which is Tuesday. But we do have lab this afternoon at 2 o'clock. So let's break now. I'm going to keep this Zoom channel open. It will stay on. It will stay active. You don't need to log out. I'm simply going to mute myself and stop my video for 15 minutes. go take a break, and we'll rejoin at 2 o'clock, okay? So I will see you soon.
Professor? Hi. I actually have a doctor's appointment today at 2:15. So, I mean, I'm probably gonna leave at 2. If I can, but by the time that I get back, I also can provide your doctor's note. But if I can, by the time I get back, if there is still time in lab, I'll definitely join in. Um, Also, is there gonna be like a video that you post later for lab as well, that I can just go in and kind of start from the beginning?
Well, normally speaking, Geneva, I won't record lecture, but I'll do part of lecture today, at least enough so that it provides some guidance for you, okay?
Okay, that'd be great.
All right. All right.
Okay.
Good,
So
good luck with your doctor appointment. Okay.
Thank you.
Okay, welcome back, everyone. So, thanks for hanging in there. So today is our first lab. And as I said, there really was nothing you needed to do to prepare for lab. On average, I will record lectures on Zoom, but I will not, generally speaking, record lab time together. Today, I'm going to make an exception to that to help a student out. But so you can expect generally lectures will be recorded, but not so much lab time. And so basically the general rhythm of how lab will happen online is that when we come together, we will take some time to review the lab from the previous week you've done. Then we will introduce a new lab activity, get you started in that work for 30 minutes to an hour so that you all feel comfortable in what it is I'm asking you to do. Give you an opportunity to ask questions along the way if you have it. And then we'll sign off for the day. You know, we're in lab for about an hour and 40 minutes. So that should give us enough time to review the previous week's lab and get a new activity started for the upcoming week. Obviously, this week is our very first lab. There was no previous lab, so there's nothing we need to review from last week. But starting next Thursday, we'll review what we're going to start today, okay? And so, as I mentioned in lecture a couple hours ago, I sent you 4 files through email, and those are the files we'll be using today. Many of those files you can just use them as you bring them up on your computer. If you so wish to work with physical copies and you have a printer, it's okay to print things out. It's okay, but you're going to have to submit your work to me online. And so if you have the option of answering questions digitally on the documents I'm sending you, or creating other information in a Word document where you organize it and label it with your name and everything's nice, right? All of these files have to get sent to me to the course Dropbox so I can see what your work's been like to help you out and to determine what credit you should be awarded for your work. So let's take a look at a couple things here. I'm going to bring, and let me say this before we get started. I'm going to say a lot of things. There's absolutely nothing today that you need to finish today. There's nothing today that you need to finish today. So don't get alarmed if you say, well, how am I going to finish that by 3:40? There is nothing today that you have to finish today, okay? We're going to get started. I'm going to try to get you to a point where you feel comfortable in what I'm asking you to do, and then we will sign off. You go have a nice Labor Day weekend, but somewhere in there, make sure this homework gets done. Next Thursday, we'll talk about it. So there's a question in the chat window. Can we print it? I'm assuming the documents I've sent. Can we print it, write it down, and screenshot it and upload it? Yes, Yasmin, but there's going to be a special way I'm going to ask you to do the screenshot. Okay, so yes, but stay with me. to make sure you understand how I'm asking for you to achieve this.
Okay, thank you.
Okay, so let's go to the course syllabus here a little bit, and I'm just going to bring it up. If you have it, that's wonderful, but I'll just bring it up for us. Okay, so here's our lab schedule, right? This week, we're going to start with something called Seeing the Invisible Leeuwenhoek, okay? And we're going to progress through the semester this way. So every week, we will review something you've done and start a new activity for the following week coming up, okay? And so, again, in your syllabus, I've placed it several times. The course drop box is here. Okay, so all you have to do is, open this. Let me open this and see what it looks like here. Okay, so this folder is empty because nobody's put anything into it yet. So it's a nice brand spanking new for everyone. You can drop your work, your completed work into this environment. Okay. All right. Now, instructions for submitting lab materials for credit, okay? Let's just go through this. Whenever you have lab materials to submit, choose one of the following options. Option A, submit work you've typed into a PDF document. Some of the files I sent you through email are already in an Adobe Acrobat PDF, and some of them might be fillable, okay? The documents I'm sending you, I did not develop myself, so I can't control which documents you can type into and which ones you can't. But what you can do is try to type into it and try to save the file under your name and see if that works. And if that works, then you can just type your information into that PDF and submit it to me at the course Dropbox URL. That would be the simplest way to go. Now, when you take a document and you save it with your name, please use a very informative description in the file name, because that's going to help me identify whose work it is and what work it is that I'm trying to take a look at, okay? So, just as an example, if your name's Sally Applegate and you're doing a lab activity on mitosis, then please make your file name S for your first initial, full last name, Applegate, followed by a description of what the lab activity was, mitosis lab. And if you have to submit multiple files, then do S Applegate, mitosis lab 1, S Applegate, mitosis lab 2, that kind of thing, okay? So make sure that your file names have your name in it and it is descriptive, okay? If I can't figure out who sent me something or what they sent me, guess what? You don't get credit for the work, and I will not track you down. Okay, so make sure you're giving me good file names. Secondly, if you prefer to write your answers out by hand, like you print out the document, Yasmin, you print out the document, you write your answers on the document, and then you want to send it to me. Okay, I don't want you to take a photograph. I don't want that. But what you could do is use one of these applications to make a PDF. These days, I think everybody has a smartphone. So you can use Adobe Scan or Cam Scanner. Install, these are free applications. You can put it on your cell phone that has a, your cell phone has a camera, and you can use these applications to physically take a picture of your work. That picture will be converted into a PDF document. That's what I want. I want a PDF document of your work. And then you can take and name it properly with a good description and your name. And then you send that to me at the course Dropbox also down here. Okay, so let me ask you in the chat window, have any of you used Adobe Scan or Cam Scanner before? Okay. So if you look through the comments, folks, you'll notice that some of you have. And what I would say is, hey, if you're having trouble, you can certainly reach out to me. But if you feel more comfortable, some of your classmates already have done this, so maybe you can reach out to them as well and they can help you, okay? Now, let me say something. I tend to use Adobe Scan just because I got started with that first in life. I became aware of Cam Scanner recently, teaching a genetics course. A lot of my students love Cam Scanner. Let me say this. I was recently reading an article on the web that was indicating that the free version of Cam Scanner might have some malware embedded in it. I didn't know that when I typed up the syllabus. So to be on the safe side, I would recommend Adobe Scan. I mean, if you have Cam Scanner on your phone now and you've been using it and you haven't had any problems with it, then it's probably okay to continue. It's just that I recently saw some news out there indicating that Cam Scanner might have some malware embedded in it. So my recommendation, try Adobe Scan and see if you can make heads or tails of it. It's pretty intuitive, okay? Okay. So Yasmin, does that seem sensible that you could make that work?
Yes, I have a printer that scans, so I can scan it directly to my computer and then rename it.
Okay. So if you have
Thank you.
another, you bet. If you have another technology solution, have at it. That's totally cool. All right. So do you have any questions at this time about where to submit your completed work or how to submit your completed work? Okay, great. All right, then let's take a look at what it is we're going to do today. So I sent 4 documents to you, and I'm just going to bring them up on the screen first to show you what you should have received, and then we'll talk about things. So you should have received, excuse me while I get my screen situation. I really wish I had like a 50-inch monitor. Okay. You would have gotten this file, which contains pictures and information of about 20 different microorganisms. Okay, so that's one file you should have received. You would have gotten this file, which is the biointeractive activity, the student handout, which lists out part 1, 2, 3, 4, and 5 on the worksheet. You also would have received an additional supplemental worksheet called Part 2. The Part 2 on the first document is referring to the second file you received. So metric conversions, units and conversions is referring to the second document, okay? So one thing that'll become clear is that to complete this activity over the next week, you will need to have some kind of calculator. And I'm hoping that if you don't already have a scientific calculator, you have a basic 4-function calculator you do to balance your checkbook at the house or use a calculator on your computer. And you're also going to need a ruler that has a metric scale on it with centimeters and millimeters. If you don't have that, I'm going to ask you to go buy one at Dollar General, at the dollar store, you know, supermarket for 50 cents. I don't think it's going to be a financial burden if you don't have a ruler, because you're going to need a ruler. So these 3 files you would have received, and you would have also received, you would have also received something else that I need to bring up here. This. You would have received instructions about this week's lab activity, okay? So before we get ahead of ourselves, let's take a look at the instructions so that what I'm saying to you makes some sense. And then we'll actually get some activity started here where you're doing things and I'm not talking. Okay, so number 1 here, obviously, your completed course lab activity materials are due in the course Dropbox. The web address for that's on the syllabus by 2 p.m. next Thursday. And that'll be true basically every week. Okay, make sure you have a good file name for it. Make sure the file name has your name and a descriptive label. Now, here's something you might be interested in. Here's questions that you do not have to do. Here's questions you don't have to do. So I'm going to tell you the things I don't need you to do, I don't want you to do, and would just be a waste of your time to do them. So, okay, so I'm telling you part 4, questions 4, 5, and 7. So what that means is on the main activity handout, part 4. Okay, so here is part 4. Questions 4, 5, and 7, you do not have to do. Questions 4, 5, and 7. So this one and this one, you do not have to do it. So you can take note of that. If you print it out, you can just put an X through it. Okay? You also do not have to do question 7. And the reason for that is because it's asking you to put it to actually make paper models and cut them out as though this is, you know, an origami class or something like that. We're not going to do that. I mean, if we were together in lab, I could organize all these materials for you and I could say, let's do it. But it's just too much, you know, since we're all spread out. So questions 4, 5, and 7 on part 4, you do not have to do. Continuing. Okay. On part 5, part 5 also talks to you about making a model. You don't have to make a model in the sense that I need you to cut something out of paper. I don't want you to cut anything out of paper. But what I want you to do from part 5 is draw a model of the cell, draw a diagram, a schematic, a picture of the cell that you've chosen, and we'll talk about that in a moment, that's magnified 1,000 times on the paper. And so that drawing that you produce is one of those things you're going to have to take a snapshot with Adobe Scan, create a PDF of it, put a good name on it, and put it in the course Dropbox as part of your completed work. Okay, so part 5 is referring to this part here, part 5. Okay, you don't have to cut anything out with scissors. You have to draw it on a piece of paper and take a snapshot of it, scan it with Adobe Scan. Okay, any questions so far? All right, let's continue. Wrong document. Here we go. Okay. All right. So additional instructions for the activity we're going to do. Okay. Hopefully, once we get into it, it'll make sense what these instructions are really saying to you. So obviously, the first thing is you're going to need a calculator and a ruler with a metric scale on, centimeters and millimeters to do this. For all the calculations that you're going to be asked to do, I want to see your work. I want to see your work. So that means you're going to have to organize some handwritten notes probably of the calculations you're doing, or you can type them out. That would be organized, but it would be more work. If you want to just handwrite it out in a nice, neat fashion, snapshot it with Adobe Scan, I'm cool with that. So for all your calculations, please show me your work, because you know what happens is you get an answer, it's wrong. I don't know why it's wrong if I can't see your workflow, so I can't help you, okay? So please show me your work and document it and submit that as part of your work for the lab. Okay, letter C. To build the skills you're going to need to do this lab's activity, you should do the parts of the student handout in the following sequence. Part 3 first, part 2 next, part 4 next, part 5 next, part 1 next. And today, we are definitely going to start part 3 in the lab together, because part 3 is a video. We're going to watch the video together in just a moment. Okay, but if you're home and alone and you say, I'd like to watch the video again, I do provide the URL to you so that you could watch it again. Okay, part 2 is going to be the extra supplemental sheet explaining metric units and unit conversions. For some of you, this is going to be pathetically easy because you're used to doing this, and that's fine. Others, it's been a real long time since anybody asked you to read a ruler, do some metric calculations. Might get a little scary there for a while, but we'll help you and get you through it. That's part 2. Part 4 is going to teach you how to calculate magnification factors as though you were actually using a microscope. Calculate magnification factors and how to calculate the true or actual size of an image that in this case is going to be on a piece of paper, right? For example, on this organism sheet, right? Here you have a rotifer. Obviously, this is a microorganism. You got a nice picture here that takes up half of a printed page. So that microorganism is not literally the size of half of a printed page. It's much smaller. Well, this scale information down here in the scale bar is going to allow you to actually measure and calculate the true size of this organism. And that's going to be part of your workflow today and through the week, is learning how to calculate the true size of these microbes. Okay. All right, and then for part 1, okay, part 1, let me show you what part 1 looked like on the handout. Here's what part 1 looks like on the handout. Part 1 says, examine the cards distributed by your teacher and follow the given instructions. Well, the cards that it's talking about is this handout with all the organisms in it, okay? So here's the instructions I'm giving you for part 1. For part 1, what I want you to do is choose any other 4 microbes from the deck of cards, that file I just showed you. Choose any other 4 microbes that you did not choose for part 5 and repeat the analysis of part 5 for those 4 microbes. Repeating the analysis will involve making information tables like this, where you take measurements, you calculate magnification factors, you calculate actual sizes, and then you determine the size when it's magnified 1,000 times. The whole point of this activity is starting to get used to units, conversion between units, and thinking about developing an intuition for the size of microorganisms. Think about whether a virus is bigger or larger than a bacterium, whether a bacterium is bigger or larger than a protozoa, so on and so forth, developing this kind of intuition. Finally, at the end of part 5 in the student handout, at the end of part 5, there is this analysis question section. Number 1 here says, which of the cells or microorganisms that you and your classmates modeled are larger than a 0.5 millimeter period? Well, for most of the time, you're going to be doing this work on your own over this following week. You're not going to have too much contact intentionally with your classmates. So what I put in the instructions for that question is follows. For the analysis question, question 1, using the 5 cells, there's 5 microorganisms you analyzed to complete part 5 and part 1, use those 5 to answer the analysis question, question 1. Okay, so just use your information that you've been working with. Don't worry about getting other people's information. Okay. Okay. So I know that might be a little bit kind of odd because you haven't had a real chance to look at things yet, but at least so far, do you have any questions?
We're going to be working on this in class, right? Like right now?
We are going to get it started together. Yes, we're going to get it started together. I'm going to try to get you to a point where you have some confidence on what I'm asking you to do. And then at that point, if folks want to log off and complete it as homework, you may do so. Anything else? Okay. All right. So don't be afraid to fire away questions, chat window. If you need something, shoot me an email, you know, that kind of thing. I will endeavor to help you. Okay, so what we're going to do next is begin the activity. And so in accordance with the instructions, we're going to begin with part 3. And part 3 is actually the video. I have a short video here I want to share with you. And so let's do that. Okay. All right. Okay. So bear with me. And here we go, folks. I'm going to pop this up and play it for us. Here we go. Yeah, here we go.
My job is to make college easier because students have a lot on their minds.
Everything that you can actually see with your eye is just the smallest sliver of life on this earth. Most of life is invisible. We still have this idea that we're the most central feature of Earth. And it's the humans that are the bystanders. The microbes are doing the work.
Wat doe je als je dingen ziet wat niemand ooit heeft gezien? What do you do when you see things that no one has ever seen before? L-A-Y, lay, u, then, they pronounce it with a V, hoek, Leeuwenhoek.
Anthony van Leeuwenhoek. He was a haberdasher in the city of Delft in the Netherlands.
And why his curiosity found an outlet in microscopes, that is just lost to history. We really don't know.
The quality of his microscope was superb. He made some 500 of these small instruments, and only a few of them he showed to visitors. He never told anyone how he made his lenses.
Robert Hooke in England, he wrote this wonderful book, Micrographia.
The first observations of the small world with lenses.
One of the first things that Leeuwenhoek did was look at things that Hooke had looked at. There was a stinger of a bee, leg, I believe, of a louse, singular of lice. But he saw some things that Hooke didn't see because his lenses were better. It was summertime, it was August. The days are so long that you get a lot of algae growth on water. He called it green clouds. Curious again, he had what he calls a glass vessel, a jar probably, and he filled it with the water. The next day, he put it under his lens, and what he saw was green streaks. Among this was all these little animals. And these things were a whole lot smaller, like 1,000 times smaller than anything he had ever seen before. And I think the line is, I confess I could not but wonder at it.
Leeuwenhoek called them in Dutch dierkens. And dierkens, that's a diminutive of the word deer.
Deer, D-I-E-R.
And which is the Dutch word for animal.
What Leeuwenhoek called them was little animals.
This was all so new. The word microorganism did not exist at the time. The word bacteria is from the 19th century.
And that strikes me as Adam in the Garden of Eden, who in Genesis named all the animals. It was just a brand new world. And he was the first person in it.
He wrote a letter to the Royal Society, one of the first organizations to practice experimental science.
And they're going, oh my heavens, what is this?
At first they didn't believe it.
Finally, the other members of the Royal Society were also able to see it. And the rest is history.
And so he discovered many things.
Sperm, red blood cells, protozoa, and bacteria.
Which nobody had ever seen before.
He is the first person to see everything he looked at for 50 years.
Van Leeuwenhoek wanted to see these things. Well, he saw them. But now we get most of life is microbial. If you look at the tree of life, only this tiny little part is every single thing you've seen. Every higher organism is covered, inside and out with bacteria. And humans would not be alive if these little 24/7 partners weren't giving us all of these genes and proteins that our own genomes don't encode. And they have all kinds of fabulous behaviors. Vibrio harvii is a marine bacterium. It looks like a sausage and it's very fast. Vibrio means vibrate. And what is amazing is that if one watches them go from a single cell to a number of cells, all of the bacteria in unison start glowing in the dark. By studying this bioluminescent organism, we discovered that bacteria can communicate using a molecular language. We used to think that social behaviors were the purview of higher organisms. What we now understand is that bacteria were probably the first organisms on this earth to ever communicate with one another. We're always looking at an unknown world. We're driven by our ignorance, and we're driven by the idea that the world must be more complex than what we understand right now. And that's enough inspiration to do an experiment. Can you imagine being the first one to see your sperm swimming around? I mean, that'd be a scary thing, right?
Okay, gang. So let me see here. Let me bring you all back here. Okay.
I just have to say, I think that's very fascinating. Like, I think we take, like, I myself, I can't speak for everybody, but have taken, like, I guess the technology and stuff that we have now for granted. Like, to be the person that actually, like, discovered that stuff had to have been fascinating.
Absolutely. I mean, what was part of his natural curiosity that he even wanted to explore this? Well, why should I look at a sample of water or soil or whatever it might be and simply look at it? It's, there's a lot of technical hard stuff that, will make you scratch your head. But when you just step back from it and look at the amazing beauty of what Mother Nature seems to have engineered over billions of years of evolution, some trial and error, it's absolutely breathtaking. So, Kit, I do see your message to me, so we should talk about that, okay? Maybe after lab today or something like that. Okay, let me just ask in the chat window. I suspect the answer to my question is yes, but I just need to ask to be sure everybody was okay. Were you able to see and hear the video? Because the last thing I hate to do is show like a 6-10 minute video and then you come back and say, hey, Dr. Allen, I could not hear a thing. Couldn't hear a thing because that's happened before. All right, so if you do have trouble with that kind of thing, please let me know. It might be I made a mistake on something, set the computer wrong, but you do have the link to it now that you can look at things. Okay, so that was the video. Kind of get your curiosity riled up about things, to share part of the wonder of discovery, okay? If this was a normal semester, you and I would be in the lab together and we'd be using a microscope. I can't use a microscope. I can't send you one, right? It's a $300 piece of equipment, can't do it. So we're stuck doing other things, okay? But we can still do some useful and interesting things. So part 3 in your student handout, just so you don't miss it, it says, after watching the video, imagine that you can ask Van Leeuwenhoek about the organisms he saw or about the ones you viewed on the card. So, you know, imagine you're having a conversation. Make a list of questions you might ask him. Try to design 2 or 3 questions that you might be curious to ask Van Leeuwenhoek, okay? And include that as part of your response with your homework today. Okay, so proceeding forward, what we're going to do is I'm going to ask you folks to start work with the metric units and conversions on part 2. Now, this will involve starting the second worksheet right here, okay? This is where I need you to start. And what I'm going to do is put you all into small groups, small breakout rooms, so that you can interact with 2 or 3 other people. And that way, hopefully at least somebody in the group has a ruler. Okay, so hopefully someone has a ruler. And at the very least, you can help each other with the math, because, you know, math is one of those topics that is so important for science, but a lot of people have phobia about it. There's nothing wrong about not understanding. That's totally cool. There's lots of stuff I don't understand. What's wrong, though, is that you give up and don't want to try, right? That's the part that's wrong. So science cannot avoid math. We got to do this kind of thing. And so I'm going to ask you to work with 2 or 3 other people in a breakout room to get started with part 2, to see if you guys as a team, you gals as a team, can work to answer some of these questions. And what I will do is periodically pop around the various breakout rooms to see how it's going. If I can provide help, I will. If you're super stuck, you can send me a message from your breakout room, say, Dr. Allen, could you please join us? You know, if I'm not with someone else, I'll come see what's going on and I'll help you, okay? So do you have any questions before I create the breakout rooms? Okay, so tell you what, I think I've given all the necessary instructions I need to give to maintain the recording here. So at this point, we're just doing the work. So I'm going to stop recording.